Thank you. Thank you also to the Fallona family to give me the opportunity to tell you about my favorite brain structure today. So in the lab, we are studying, we're trying to understand the human brain, and we're trying to understand especially how the human brain produces movements. And the one single brain structure that we're mostly interested in is the human cerebellum. Now, the cerebellum is this very folded brain structure in the back of your head. Cerebellum is Latin and stands for the little brain, and it's tucked very safely underneath the big brain, the telencephalon, which in the human consists mostly of this big neocortex here. Okay, and it's kind of subdued basement location might be one of the reasons why it's often ignored in neuroscience. So many of my colleagues, tell me what they think the main function of the cerebellum is basically to hold up their brain structure that they study, namely the visual cortex and the parietal cortex here. Okay. But what I would like to do today is take the opportunity to take you on a kind of slightly historical journey to give you or convince you that the cerebellum is actually one of the central places in the human brain that are really essential to understanding a lot of things, a lot of different behaviors. And it's also an opportunity to kind of highlight, in the spirit of this award, the interdisciplinary science that is behind it. So you'll see that there are many influences from very different parts of the science that have brought us to this place of understanding the cerebellum as we understand it now, but also into the future to understand it. And this historical journey, well, we should really start with the old ancient Romans, which already kind of recognized that there was a cerebellum and named it, but we might as well start with the early European physiologists. And so I'm going to start with this gentleman here, Franz Josef Gall. He's known as the inventor of phrenology. So this is kind of the science where people start, rather than actually looking at the brain, they looked at the skulls and tried to infer from the bumps on the skulls that if you have a bump here or there, then maybe the underlying brain structure is really big. And then Franz Josef Gall associated certain faculties or characteristics with those bumps. It wasn't a very exact science. It wasn't very empirical. And so he was mostly guided by his intuition. And if you're a pre-Victorian scientist, what could you possibly hide in the brain's basement? Obviously, it can only be the most depraved and vile tendencies. So he convinced his contemporaries that the cerebellum really is associated with reproductive drive. Okay, that's what's down there. All right, and so we're kind of shaking our head about it now, and it certainly didn't turn out to be true, but it had a long impact on society and on science and on medical writing. And so even in Victorian times, there were these character guides that were published, right? Where warning young ladies when they looked for a husband, they should be aware that not to pick somebody with a bump in the back of the head, because that is predictive of infidelity. Okay, and they should instead rather look for husbands that have bumps like this, that have these enlargements in these more enlightened parts of the human brain. Now you're laughing now, but look at your partner very carefully, and don't tell me I didn't warn you. Right. Now. We laugh now, but obviously this time had many scientists that did much more careful empirical work. And I just want to highlight here Pierre Florence, who actually worked with animals. He worked with chickens and pigs because that's what he has access to. And he very carefully lesioned the cerebellum in these animals and found and wrote in his writings that the slightest disturbance of the cerebellum alters the harmony of coordinated movements, and complete removal of the cerebellum abolishes them completely. So he was right on the money there, right? So when that is kind of the main symptoms that you find with cerebellar lesions in animals. You see disturbance of not necessarily that the movement is completely plegic like you would get in a cortical lesion, but that the harmony of movements, the coordination of movements is disturbed. Now, this is also the classical clinical picture that you read in neurology and neuroscience textbooks. And a very keen description of this picture we owe to this gentleman here, Gordon Holmes, who is an Irish-British neurologist. And he was appointed to be consultant neurologist to the British Army in the First World War. And the reason he got interested in the cerebellum had to do with a design flaw in one of the key military equipments that was used on the battlefields back then, and that is this Brodie helmet. It's kind of a British patent. It was produced in Britain, and it was worn by both British and American troops, and saved many soldiers' life because it protected the head from bullets and shrapnel. The design flaw was that it didn't have neck protection. Okay, so many soldiers actually survived these gruesome battles, but at the same time they carried away lesions to the back of the head, often a bullet or shrapnel piece either on the left or the right side of the head. And so Gordon Holmes had the opportunity to study many veterans that had a one-sided cerebellar lesion. What he found is that if you have a left cerebellar lesion, you would show motor deficits on your left side. So this is slightly different from what we know about neocortical lesions. So if you have a stroke on the left side of the brain, you're going to be plegic on the right hand. So the right side of the brain controls the right hand, but actually the left cerebellum talks to the right part of the neocortex and controls the left hand. So things are double crossed in the brain. Now, Gordon Holmes also very carefully characterized these movement deficits. And so these are some of the first kinematic recordings that are published from most neurological patients. And what he did was he put a flickering light source. He didn't have video cameras or anything, or motion trackers. So he put flickering light sources on the tip of index fingers and then used long exposure photography to get these beautiful kinematic traces. And so what this is a trace of a patient with a left cerebellar lesion drawing a square in the air with his right hand, so you can see that looks perfectly normal and fine, and then attempting the same thing with the left hand. You can see that there's quite a bit of tremor there. Okay? And this tremor is slightly different from what you see normally in Parkinson's disease. So in Parkinson's disease, you see tremor, but it's what's called a resting tremor, like it occurs when you don't move. Cerebellar patients are fine when they don't move, but when they start moving, then they start shaking. Okay? Now, Gordon Holmes also spearheaded the standard neurological test to bring out cerebellar disorders, and that is the finger-to-nose test. You stretch out your finger, and you have to point to your nose. Okay? So that's, and let me show you what a cerebellar patient looks like when he tries to do it. So I'm going to play the video, and I hope the sound is on. If it is, then also try to listen to the speech of this patient here.
Now what I want you to do is take that right arm and touch your nose again with it. Okay, do that again. Okay, let's see you try to go a little bit faster this time. Okay.
Oh, when I go fast, I lose everything.
It's a little dangerous, doesn't it? Okay.
So this patient has cerebellar degeneration, and yeah, it's unfortunate. He's instructed to do the finger-to-nose test. He's holding out his hands fine, and then he's pointing, and you can see it already that he points to his hand, and then he's asked to point faster. And when he points faster, you really see that he becomes completely discoordinated. And so this is a symptom that that is called ataxia. That is really the loss of coordinated movement, which is one of the main symptoms in cerebellar disease. Unfortunately, I can also ask my audience, what did you notice? What else did you notice? And what you notice that the patient speaks actually in this video, and he speaks like this, right? I mean, he loses the fine articulation, and that is another symptom of cerebellar disease, which is called dysarthria. And then there's the third symptom that you can't see because they're sitting, that is often these patients lose balance, and they have a very broad-based gait and kind of lumbering gait to stay stabilized. Now, those three symptoms together, does it remind you of anything? Being drunk, exactly, yeah. So these are the reason for that is that these motor parts of the cerebellum, the Purkinje cells, are extremely sensitive to alcohol, okay? And they really shut down their function once you have a certain level of blood alcohol. Now, there are other parts of being drunk that you can't blame on the cerebellum, namely kind of the social disinhibition and so on. So that's prefrontal cortex fault, likely. But those three symptoms are very clearly cerebellar, okay? So next time you have some alcohol, and you deactivate at least one crucial part of your cerebellum. Okay, so this is kind of also today what you find. These three symptoms are kind of the key neurological textbook, neuroscience textbook, deficits in cerebellum. At the same time, then after the Second World War, we also had influence from biology and neuroscience in that we really started understanding the cerebellar circuit at the microscopic level. And when you take a picture of a slice of through a cerebellum, what you see is that the cerebellar cortex looks very different from the neocortex. It consists of these very large cells, here yellow, they're marked yellow in this picture, and they're called Purkinje cells. And they have these enormous dendritic trees that form what's called the molecular layer, and they kind of sit on a bed of rice, and that rice is really kind of like sushi, is the granular cell layers. Now, the granular cell layers are very, very small neurons, and you have a lot of them. Indeed, about 60 to 70% of your neurons in your brain are cerebellar granular cells. Okay, so there's the most frequent neuronal type in your brain. Okay, and the reason is that they're so incredibly small, and you have incredibly many of them, 50 billion roughly. Okay. Now, these Purkinje cells are absolutely remarkable. They're remarkable already in the mouse, and this is the mouse preparation, but they're even more remarkable in the human brain. Okay? This is the very early drawing of a human Purkinje cell from 1899 from Cahal, which you might have heard of, and he drew this Purkinje cell. So there are these big dendritic trees, and these dendritic trees that's kind of the input into this neuron. This is where they receives all the input, about 1 million synapses on the cell, and then these inputs are integrated in the cell body, and then the Purkinje cell computes something and provides one single output. So this is kind of the computational unit of the cerebellum. Now, the interesting thing about these cells, they're very unique. They look very different from neocortical neurons in that they are absolutely flat. So they're these big 2D sheets. They're not like the normal trees that you see. They're these sheets. And these sheets are in the cerebellum. They are arranged not like an old growth forest like in the neocortex. It's all kind of messy. They're very orderly arranged. Okay? It's looks like a fruit orchard in a way. All these Purkinje cells stand in one direction. Okay? And then there are these granular cells that sit underneath, and these granular cells give rise to these parallel fibers. And the parallel fibers run exactly orthogonal through that lattice. Okay? So it's an arrangement that has maximized the number of parallel fibers, the number of inputs different inputs you can give to a Purkinje cell, and the number of Purkinje cells that these parallel fibers can achieve. So it's nearly as if an engineer had come up with this, right? So it's very, very different from the more chaotic organization of the neocortex. So it's this beautiful arrangement. And we also understand now how this arrangement actually, what is a compute? What could be a basic computational principle? And so we need to kind of review how inputs and outputs are going through the cerebellum. I'm going to just walk you through it real quick. So the neocortex, your brain, sends about 40 million fibers down to the spinal cord and the brainstem. So this is all the information that leaves your brain to control your body. Okay? So 40 million lines of information in a way. 90% of that output goes to the cerebellum. Okay, to the pontine nucleus, and then is multiplexed on these 50 billion granular cells. So it's this big information expansion. They send out the parallel fibers, as I said, and then there are these Purkinje cells that listen to 150 to 1 million of these parallel fibers, and then provide one single output, each of them. Now, the unique thing is also that they receive one single axon, one single information line, the so-called climbing fiber. And this climbing fiber is a very sparse signal. These climbing fibers spike maybe once or twice a second. And the whole life of a Purkinje cell consists of trying to predict when that climbing fiber input comes. Okay? So that's what it's made for. And so what they do, these Purkinje cells, they wait, and they try to listen to this parallel fiber input, this weird big complex matrix, this pattern that they get. And from that pattern, they try to predict when the next climbing fiber input will come. And then, about 50 to 100 milliseconds before they feel that climbing fiber might spike, they're gonna scream, lower the firing rate, so disinhibiting the deep cerebellar nuclei, that then goes to the thalamus and back to the neocortex and says, hey, something is gonna happen. Okay, so it's this predictive mechanism that they're building. Okay? At the same time, because it's a good reinforcement or like error-based learning mechanism, it also cancels its own error signal. So it kind of tells the error, the source of the error signal, so-called inferior olive, tells like, don't tell me that there's a climbing fiber coming. I predicted it. So when it can predict the climbing fiber, it cancels its own input signal, and that reaches some equilibrium. So this is really a really good description of what the circuit does. There are many details to that, but this idea was already proposed in 1969 by David Ma, and then refined later by James Albers and Masaharu Ito. And so really brings home the idea of the cerebellum as a machine that has this big, where each Purkinje cell has this big matrix input and learns to predict a very sparse input signal. That's kind of the computational description of the cerebellum. And so I think if there's ever one single Nobel Prize in neuroscience or in physiology missing, then it's that one, the Ma-Albers-Ito theory, because it's a theory really proposed. I mean, the original papers are brilliant, and it's this theory really has stood the test of time. It's more than 50 years old, and in the big picture, this theory stands today and leads our research. Okay, so we have an idea, we have a very good description of the clinical picture of cerebellar disease, and we also kind of know what the circuit computationally is doing. How can we link the two? And this is where another scientific perspective comes in, and namely from control engineering. You can ask any first or second year engineering student here at Western to how do you control a plant or a body? Well, we will tell you, you need a controller, you need some input, some sensory feedback or state estimate from the thing, you need a goal, and then you can send out some commands, motor commands, and you can control that thing. Okay? Also, every engineering student knows if there's a delay in the loop, so if you're trying to control kind of an extraplanarity vehicle or something, or the body, in the body the delay is roughly 100 milliseconds from the time that you send out motor commands until the sensory feedback comes back that tells you that have you moved. It's troublesome to do fast control. Okay? And the reason is very simply, if you try to move your hand to your nose, to a goal, the controller will just push towards the goal as long as it thinks that the hand isn't at the goal. So it's going to move the hand to the goal. But at the moment the hand reaches the goal, the sensory feedback will be still 100 milliseconds delayed. Okay? So the feedback will still tell you that the hand is actually not at the nose yet. And so the controller, with a simple control policy, will push further and overshoot the nose. By the time it's realized that the hand is past the nose, it will reverse the movement and go back. And then overshoot again, and then so on, so on. And you see exactly what you didn't see on the video, the typical cerebellar ataxia. Okay? So Daniel Wolpert and Chris Mayall realized that this would be possibly a good explanation for cerebellar ataxia. Okay? And they also proposed, took inspiration from control engineering to explain what the cerebellum is doing in the circuit. What a control engineer, what people are doing there is to build a forward model. So a predictive mechanism within this controller that simply takes a copy of the efference command, okay, and it's a model of the body. So it tries to predict from the motor command and what it knows about the body or the plant that it's trying to control, what's going to happen when these motor commands are actually executed. Okay? And so it can do this because it's internal feedback loop much faster than this delayed feedback loop, and so it can be predictive. Okay? And so that basically leads to an alignment of the sensory feedback to the current where the hand is currently. So it can kind of tell the controller, based on our sensory feedback and based on our prediction, this is where we think the hand is now. So it kind of gets a good state estimate, and then the controller actually turns out to do smooth movements. And these mechanisms are built in the first application was in industrial applications to roll steel, where you had to kind of adjust the height or the pressure on the steel sheet to get the right thickness. And there is a lot of time delay between these. And so this is where this invention came first in the 50s. And now it's being used in many control applications and robotics applications. Okay. Good. So far, we have a pretty compelling story. We know the cerebellum is involved in coordination of movement. We have a good clinical deficits. We have a circuit description. We even have an engineering picture of how this all things together. Now, the fact is that this is not where the story stopped, and the story becomes in the end of the 90s much more complicated. And again, this comes from yet another scientific perspective, and that is evolutionary biology. Okay. So even insects have cerebellar-like structures, but a proper cerebellum really only comes to the fore when fish develop jaws and become predators. Okay? So when they develop big bodies and need to do fast coordinated movements, and so sharks have a very pronounced cerebellum. And since then, in all vertebrae, the cerebellum has kind of developed in line together with the telencephalon, the big brain. Okay, so if you look across it, there's always similar ratio of these two structures. Now there are some notable exceptions of that, and I think this is just a fun one that I like to show. Because there's a family of fish which is called the marmorets, where the cerebellum has grown to about 5 times the size of the big brain. So now the cerebellum is a big brain. It kind of looks like this. This is how this family of fish looks like. And so basically here the cerebellum is so enlarged that it's now sitting on top of the telencephalon, whose function is it now to hold up the cerebellum. Okay. Now, why do they have such an enormous cerebellum? Well, first of all, we have to disappoint our friend Franz Josef Gall. Their sex life is pretty ordinary. There's nothing special here. But these are electric fish. So they're weak electric fish. They send out electric signals to navigate in the muddy waters, in the muddy tropical waters that they live in. And it turns out that one big part of their cerebellum is dedicated to the analysis of those signals. And this forward model mechanism is very handy here. So basically what this fish is doing, based on the signals that it sends out, it makes a prediction of the signals that it ought to get back if there was nothing in the environment. So that subtracts basically out 99% of the signal that it gets back, because that's just predictable by the body position and the signals that are being sent out. And the fish or the nervous system can focus on the violations that would indicate that there's an object, an obstacle, or predator in the environment. Okay. They also have exquisite trunk control. So I encourage you to go on YouTube and type this in and look at like how they rummage under stones and so on for food. So, and a part of the cerebellum also does that. Now, so far again, we are very consistent with the story of a forward model for motor control. Now, the break comes when we look at primate evolution. So as you know, we have evolved from primates, and one of the big differences between us and lower primates is that the neocortex has enlarged tremendously. Okay, first in big apes, chimpanzees, and so on, and then to the human. Okay? And this evolution or this enlargement of the neocortex is mostly driven, it's not driven by motor structures, it's driven by your prefrontal cortex. So this is an area that is involved in abstract thought, and your parietal cortex, which just kind of has spatial functions. Now, if the cerebellum was really for motor control, you would say, well, it should just only scale with the motor regions that haven't really scaled up that much. But the fact is that the cerebellum has totally kept pace with the development of the neocortex, and maybe even in great apes outpaced it compared to lower primates. Okay? Even in more recent human evolution, if we compare the skulls of Neanderthal to the earlier Homo sapiens, okay, what we find is that the prefrontal cortex was actually bigger in Neanderthal. The parietal cortex was bigger in Neanderthal. The one place where the early Homo sapiens had an edge over the Neanderthal was the posterior fossa, or the cerebellum, in the skull, up of brain. Okay? So that is the one place where the early Homo sapiens has a bigger brain structure, and that might have allowed it to outcompete the Neanderthal, as we know. Okay. So this is a cerebellum, human cerebellum, post-mortem. This is a picture we took at UCL at 150 micron resolution in a scanner. And I'm just showing this to show how incredibly folded this human cerebellum now is. So you see here, very fine, the molecular and granular cell layer, very tightly folded. You see these big dentate nuclei, the big deep cerebellar nuclei. And based on this scan, Marty Serino and I created kind of the first full unfolding of the cerebellar cortex. Oh, that video is working. So you can see, using computational anatomy, we reconstructed the surface, and it took Marty about 6 months of careful programming and handwork to really being able to inflate the surface and then stretch it out into a flat sheet. So it turns out that the surface of the cerebellum, this highly folded surface, is about 1.2 meter long and 15 centimeters wide, and it's just folded like an accordion in this one direction. And that has to do with the parallel fibers, because the parallel fibers run up and down, and that gives a stiff manifold that you can't bend anymore. So it just folds in this one direction. Okay. Now, we also found that it is about 80% of the surface of the neocortex. So, it's the little brain, but it's not that little. And many people before us had pointed out that the parts connected to the cognitive areas are really the ones that have enlarged with evolution. So in the 80s already, Leiner and Dow then asked the question, well, might it be that in the humans the cerebellum isn't just a motor structure, maybe it also contributes to cognitive function? And this is something that really has revolutionized in the cerebellar field since 2000. And another reason for this revolution is the advent of functional imaging. So in the end of the 90s, really functional imaging took off, and at Western we're of course one of the special centers here in Canada for imaging. So we have a 7 Tesla magnet. You can put people into the scanner like this. You can get a very accurate anatomical image of their brain, but you also can get these much more fuzzy looking functional images. And when you and that only takes a second roughly to get a full snapshot of the human brain. Okay? And if you just look very carefully at that movie, there's a movie I'm playing. You might not notice this, but there's a little flickering right here. You see here, there's a little artery, and you can see that pulsing. So this is actually the blood, the heartbeat or the pulse that pumps blood through this part of the brain. So this scan is actually sensitive about how much blood is currently flowing into that particular pixel. And so we can look at these pixels, and we can look at their brightness, and we're going to find that their brightness is varying. And when the brain area becomes active, when the neurons are starting fire, more blood rushes in, and that we can pick up with functional magnetic resonance imaging. Okay. So this is the main technique that my research group is using to study the cerebellum. And so what has been found since the late 90s is that not only motor task activates the cerebellum, a lot of different cognitive tasks that don't involve any movement at all also activate the cerebellum. Now, when I started with it, this evidence was still rather patchy. And the reason is, like good neuroscientists do, the neuroimages also ignored completely the cerebellum, to the degree that the standard analysis packages, so this is one written at UCL that, at that time, about 90% of the imaging studies used to analyze it simply cut off 40% of the cerebellum, right? It only analyzed data in this blue bounding box, and then that was ignored here. Okay? So if you look at the literature, you have really a large missing data problem, and it's not missing at random, right? It's very discriminatorily missing against the cerebellum. So when I started with this, the first thing I did was improve this, and we took pictures of human cerebellum. So you can see while the structure of the human cerebellum looks very similar across people, the shape looks a little bit different. So this person has a very happy and smiley cerebellum here. And this one has rather a droopy and sad one. But the structure is all there, and these are healthy individuals. And so what I developed was an algorithm that can morph those images and try to get a representative shape template for the cerebellum. And this turned out to be a really good idea, and is now a kind of the standard in the field for cerebral imaging, and we're supporting this toolbox to today. And we also developed this flat representation of the cerebellum. So this is basically, we just reconstructed the outer hull, and then we can inflate this. This is just for visualization of functional imaging data at this low resolution. This is not the real 1.2 meter long surface, which will also be very unhandy in a journal, right? Because you have these very awkwardly formed figures. But this is, remember that behind this flat map is really one that is 15 centimeters wide and 1.2 meter long. But it's a very important visualization tool, and again, it's widely used in the field. And we also generated atlases that are now widely used in the field to separate the different lobuli, the big anatomical subdivisions that you can see in the cerebellum. Now, when we started, as I said, the imaging data was rather spotty in the cerebellum. So we decided that we really needed a ground up. When I started here at Western, we decided we really needed a ground up study of the cerebellum. And so Maeve King, who was a master's student at that time, and I started developing a task battery where we just put people in the scanner, kept them there for 6 hours, and let them do all kinds of possible tasks, up to 50 tasks or so, task conditions. And what you can find is that when you move your right hand, you get the right cerebellum. If you move your tongue, you get this tongue region. So when you move your right foot, you get this region. You can contract your glutes. There's some representation there. You can eye movements. So these are the normal motor structures of the cerebellum that you can map. You can also activate the cerebellum very well with these kind of working memory and intelligence tasks, right? So things you would find, spatial map, finding a path through a labyrinth or mental rotation or so, that activates. Doing some arithmetic math problems also activates the cerebellum very well. But what we also found, inconsistent with other literature, is that purely resting in the scanner, just doing nothing, okay, watching other people do things like tying knots or watching a romantic movie or thinking about other people, right, that also activates these other distinct parts of the cerebellum. Now, the problem with the field is that people have summarized these data often in terms of the lobuli, because that's what you can see on the anatomical scan. And it turns out when you look at these activation patterns, that these activation patterns do not really respect these lobular boundaries. So you can see here watching a romance movie leads to this activation in this zone here that just crosses these boundaries that are shown here in white lines, but doesn't activate the entire lobulus, right? So it kind of like activates these medial to lateral zones. And so we wanted to really generate a functional atlas. And so this is one of my computer science graduate students in the lab, Da Chi. He just defended and is now starting at MGH in Boston. And he developed a way of taking many different data sets from the literature that we could get and build a model where you can fuse that data into a single map. And so here basically, the problem is that these data sets are of course different. They have different number of subjects, different signal to noise. So he started with building a data set specific model, probabilistic model for each of these data sets, and then linking them together in an hierarchical fashion with a group map. And so that's to make the computation feasible. There was a lot of trickery involved. But ultimately what we did was develop a kind of consensus atlas of the human cerebellum based on about 700 tasks. Okay, and so this is really teamwork from the lab, but it was led by Caroline Nettenkoven, who sits in the back there, a fantastic postdoc. And so just submit it. And so basically what you see here, this color scheme is two regions that have relatively similar color also are activated by similar tasks. Okay, and so what you can find is the normal motor regions. You can find regions that are involved in action observation. You have these regions that work in working memory, and you have regions that are much more involved in the social linguistic domain. How much time do you have, Paul? How much? Oh, okay. 11?
Like 15 minutes?
15 minutes, okay, perfect. All right, good. So basically, so we now have a very comprehensive map of the human cerebellum, and in agreement with other literature, it shows these maps really show that 70% of the human cerebellum isn't really activated by motor functions. It's really involved and activated by more cognitive functions. And so this really poses a big puzzle, right? And the puzzle is that if you have a lesion in adulthood in the cerebellum, there are some cognitive deficits, but they are very mild. So those cognitive deficits involve some working memory problem, maybe some problem in verbal fluency, maybe some soft hints of executive functions. But the patients that come to the neurologist's office, they don't complain about these things. They complain about the fact that they can't walk anymore, right? And that the eye movements are screwed up, okay? So that contrasts, however, with the consequence of cerebellar damage during birth or in early childhood. And that actually has quite profound differently consequences, and it delays the development of social, linguistic, and cognitive functions quite tremendously. And one of the prime examples for that is autism. Now, this is a review paper. It's a little bit old, but as you know, autism is a highly genetic disease. It's, if you have an autistic identical twin, you have a very largely increased risk of also being autistic, more than 50 times. There are some environmental factors, and those environmental factors here are dedicated in the dark blue bars. Okay, so for example, maternal stress during pregnancy, like a predator flying around, increases the risk of an autistic kid by threefold. Okay. Even after factors after birth, like social deprivation after birth, has a really high risk factor. So this is from a study from kind of notorious Romanian orphanages who basically isolated their kids, and they found an 8 times increased risk of autism developing. But the highest, from all the environmental factor, by far the highest factor is this bar here, and that is a cerebellar lesion, a small cerebellar hemorrhage at birth or shortly after birth. Okay? So that is the biggest risk factor environmentally to develop autism. So this kind of led to the hypothesis that maybe what the cerebellum does to human cognition in adulthood might be rather soft and modulatory, but its main function is to set up these neocortical circuits, to teach the neocortical circuits the right setup in development. So this also kind of leads now to where our lab wants to go in the next year. So the one thing about, if you have a kid and you're going to a pediatrician and say, is my kid healthy? The first thing that they do is they measure your kid, you can get the weight, get the height, and then tell you your kid is in the 5th percentile of weight or something for that age and that gender. Now, shockingly enough, and this is called a growth curve and has been, it's kind of one of the standard tools in pediatric medicine. Now, shockingly, that doesn't exist right now industrial strengths for brain structures. Okay, so many people are working on this, that you just can take a brain scan and say, here are the percentiles, the age-appropriate percentiles. But again, the cerebellum is being ignored, and so we just got a $1 million grant from the RENA Cerebellar project. So this is a private funder from Texas to develop exactly those growth charts. Okay, so the goal is really to use open data and develop these growth curves. Also to make them longitudinal, which is kind of an interesting statistical challenge, because having a child that might be in the 5th percentile for a certain brain structure or a certain part of the cerebellum might not be that alarming. That might be fully normal. But knowing that this child 2 years ago or 1 year ago was in the 75th percentile and suddenly dropped, that is an alarm sign. If it tracks a percentile, maybe that just still constitutes normal development. So there's a lot of added information of every repeated measures of the same child. Okay? And so that data is now starting to be available, and that's what we want to look at. Now, here's a figure, kind of slightly faint, of our first study in this respect, where we show that these motor structures of the cerebellum mature actually very early. So they are nearly fully formed at age 5. But these cognitive areas, they keep developing into teenhood. Okay, so they keep developing probably into the 20s. Okay, good. Another thing is that we are working on is that we have now a group map. That's great, but there is a lot of inter-individual variability. So every brain is organized differently, and looking at individual brains is really important for clinical diagnostic. Okay, if we want to do something like precision medicine or use that for prediction. So if you scan people like we did here for 5 hours, say, you get very good individual maps, and you can see that the organization of these functional regions is quite different across people. Okay. Now the question is, well, in the clinic you don't have 5 hours with the subject. You can't just pay 5 hours imaging to make a diagnostic. So we need some way of using something like 10 minutes of data, which is often kind of the standard in a clinical study, to get the snap. And so how do we do this? Well, again, because we've developed a Bayesian approach, we can now use our group map as a prior and get an optimized estimate that integrates the two. And we can basically get, from 10 minutes of data, a map that is roughly as good as maybe 30 to an hour of scanning on the individual subjects. So that's a big advantage. And we think this will be incredibly useful both for clinical science and maybe also in clinical diagnostic at some point. And then lastly, of course, we really need to understand how that cerebellum talks to these neocortical regions, because that's really the critical thing that sets up these circuits. And so we're building these mathematical models to understand how these two areas are talking to each other as global structures, what develops normal development, how does this develop normally, and what of these patterns are predictive of dysfunction. So this is where we're going. And I want to acknowledge a lot of the amazing trainees that have worked on this. I kind of mentioned many of them along the line. I want to acknowledge my collaborators, especially Richard Ivory, who was my PhD supervisor at Berkeley, and I still work with him. And the funders, and I, of course, want to thank the Fallona family for their generous gift to Western. Okay, thank you.
Okay, so Jörn, we'll take some questions. I guess, Sarah, we've got a bit of a window into what's happening at the grad club on Friday, eh? Huh? And maybe, Jörn, if you don't mind, just to repeat the question into the microphone.
Sure.
Okay.
Yeah.
So Sarah has a question.
I have a question.
Yes.
So if the cerebellum is contributing to some of these cognitive functions, does it add resilience if you get an injury in the upper story of your brain?
That's a really good question. So yeah, so that's, what is it, there are actually some people that are working on ways of recovering cortical stroke by stimulating the cerebellar deep nuclei. And that actually seemed to have some first indication of success. So just by driving the excitability of these cortical regions, the cerebellum provides a very powerful excitatory input to the neocortex. And so just by putting a stimulator into the output nuclei of the cerebellum and kind of driving the signals, you can actually seemingly accelerate stroke recovery in the motor cortex. So it's a really good, but this is a very new idea, and we don't have much data on that yet.
Okay, so we have one here online. Can you say more about how cerebellum trains cortical areas? Do we know of specific predictive models in neocortex that are trained by the cerebellum?
That's a good question. No. No, I don't know. It's a complicated issue. I wish I did. That would be the next Nature paper, right? The problem is that prediction, obviously, also the neocortex, the human brain is just generally predictive, right? That's why we have these big brains, right? So prediction is obviously very good because you can adjust your behavior before your enemy can, okay? Predicting where that predator is going to go, saves your life. So there's highly selective pressure for this. And so a lot of what the brain, also the neocortex is doing is prediction, okay? So now how does cerebellar prediction, what does it add, right, to these cortical predictive circuits, and how do they interact with each other? And these are all fascinating questions which we don't have the answer to right now.
Okay. Yeah. Other questions from the audience?
The discussion of the effects of alcohol and then the effects on childbirth made me think of fetal alcohol syndrome. I was wondering, have you looked at that, or have you looked at other, as well as looking at autism, other possible?
Yeah, so the question is how fetal alcohol consumption during maternity influences the cerebellum. Now, it turns out that nearly everything that happens during the pregnancy, the cerebellum is one of the structures that starts developing very early, and then kind of hangs out for a little bit, and then really in the last trimester ramps up again in development. And it's very sensitive to perturbation. It's very sensitive to stress. It probably, I'm not an expert on that, but it's probably also very sensitive to alcohol. It's very sensitive to other drugs as well. Okay, so there are a lot of problems that we see or that the pediatricians see. And one of the big risk factors for cerebellar hemorrhages is premature birth. So if you're born before 30 weeks or 32 weeks or so, you have about a 10% chance of having a cerebellar hemorrhage. And for some reason, and I don't know that we know the reason right now, those hemorrhages, bleeding, happens in the cerebellum, not in the neocortex. We don't know why. And so that's a huge risk factor. Morphine that is given to premature babies after they leave the womb to manage pain has a really disastrous effect on cerebellar development. And so pediatric medicine in Canada is now shifting away from giving morphine to these little babies. So there's a lot of sensitivity in how the cerebellum develops. Yeah, it's kind of a delicate flower early on in development, much more than the rest of the brain. Yeah.
Okay, there's one more here online, and I'm going to have to sound this word out. Are there cytoarchitectonic or other structural differences that map onto the functional subdivisions?
Oh, that's a great question. So traditionally, so in the neocortex, you might know that early anatomists have found that different parts of the neocortex, when you slice them up, put them under a microscope, they look different. So this kind of Brodmann kind of built these nice atlases. In the cerebellum, that circuit that I showed is kind of uniformly preserved across the entire cerebellum. It's very uniform. So that's kind of a puzzle. Why, so there aren't really strong differences in the circuit itself. Now, the more we look, the more slight differences we find. So there are some medial to lateral zones if you stain with certain enzymes, the zebrin zones, and also Purkinje cells in these different zones behave slightly differently. And so part of the work that we are planning right now in collaboration with MGH and Amsterdam is to try to map that in vivo using imaging and trying to see if we can map those zones. But if these zones exist, we don't know where they are in the human. We have very good in monkeys and rats and mice. We have good maps, but not in humans.
Okay, any other questions from the audience? Here.
Why does the cerebellum has like 8% of surface area, compared to the cerebral cortex, but it gets a small body?
Oh, the surface is much thinner. So the cerebellar cortex is only 2.5 millimeters thick, and the neocortex can be about 5 or 6. And it's like the neocortex has also much more white matter. So it's just a much more thin, flexible surface. It kind of stretches out also on these very narrow folds. It's kind of more like it's kind of like a washboard, kind of not like a homogeneous surface, but it has these kind of clump structures. So it's much more tightly packed. Yeah.
So, how do you get children, especially the very young ones, to stay still for imaging so long?
Okay. So Emma left, right? So I don't do this work. I haven't worked with little kids. That's a good question. So it's interesting. So neonates you can actually put in the scanner. They fall asleep. So that's fine. They like if you had a baby, that like the more you rock them and the louder it is, the more they fall asleep, right? So that works very well. But then there's a window between age maybe 1 and 4 where it's nearly impossible, very hard to scan them and have them lie still. I mean, you don't want to sedate them. Okay. After age 4, they're kind of like, ooh, I'm an astronaut. I can go in the scanner and then play tricks like this, and then that works. So there is actually, when you look at the data, we have a gap of data between age 1 and 4, and that just hangs. I mean, there are people who do this in various groups. Also, here at Western, Emma Durden does work in that age range. I don't know. I mean, I admire them that they can do that. But yeah.
Okay. Last call. All right, join me in thanking Jörn for a really great